So we're going to change directions a little bit and start talking about cell membranes. It doesn't mean we're not going to be talking about proteins, because proteins make up 50% of the cell membrane and they carry out so many of the activities that a membrane carries out, like transport and cell adhesion, receptors that recognize signals from the surface. And so we're going to talk a lot about proteins later, but I want to start just basically talking about cell membranes, and in particular we'll talk about the plasma membrane, which is the membrane that surrounds the cell. So we're going to look at membrane structure, we're going to look at membrane function, and also the proteins. And what this just shows here is a classic drawing of a cell and showing all the various membranes. The plasma membrane, of course, the Golgi apparatus is composed of membranes. The mitochondria has outer and inner membrane. Transport vesicles have membranes. The nucleus has a double membrane, and one of those, the outer membrane, is connected with the endoplasmic reticulum. So lots of membrane structures in the cell. And of course, the membranes are based on the lipid bilayer, which we'll talk about in a second. But I referred to that very, very early at the beginning of the semester when we talked about hydrophobic and hydrophilic interactions, and we looked at the fatty acids and the phospholipids and how they can arrange themselves due to the interaction with water, where the hydrophobic region or the nonpolar region of the amphipathic fatty acid turns inwards to exclude water, and so you end up getting this lipid bilayer, which basically is the structure of the membrane. But we want to look at, particularly you want to look at the plasma membrane. And what I have written on the right-hand side is some of the things that are important. Defines the outer boundary of the cell, maintains the integrity of the cell, interfaces with the environment, which is extremely important, and that's what is actually shown in that little drawing at the bottom. It shows that through the membrane we receive information from the environment. That's important. Holds tissues together, controls transport in and out, and that's also shown at the bottom, number 2, import and export of small molecules. So it controls transport in and out and participates in movement, and that's also shown at the bottom in red. Capacity for movement and expansion. They can some cells can crawl along a surface. So the membrane has extremely important functions for the cell. A lot of those are carried out by proteins. But let's just look at terms of the simple drawing or simple presentation of what a membrane looks like. So this is just a simple presentation of a lipid bilayer membrane. And of course the lipids are amphipathic, and so the polar region is exposed to basically aqueous solution or water-based solution, which would be the exterior of the cell, the interior of the cell, the cytoplasm. And of course those lipids arrange themselves so the interior of the lipid bilayer is hydrophobic and the exterior is hydrophilic. So 50% of the membrane is lipid, based on lipid. 50% is actually protein, and the proteins are what carry out so many of the functions. Now, if we talk about just strictly the lipid bilayer, that's where I want to start with, but there's not a lot to really say about it. At least I don't want to spend a lot of time on that aspect, but the lipid bilayer is made up of 30% phospholipids. We'll look at what those look like in a second. 30% phospholipid, 10% glycolipid, that means there's a sugar attached, and 10% cholesterol. So that is kind of the simple view of the lipid bilayer. Let's just look at what these lipids look like. So these are the 3 classes of lipids that we see in the lipid bilayer of the membrane. All of them are amphipathic, meaning they have a nonpolar hydrocarbon tail and then a charged or polar group, which is shown at the top in blue. And if you look, first of all, at the phospholipids, this is kind of just one of many phospholipids that are found. So they have two hydrocarbon tails. They're connected by this 3-carbon moiety, which actually is glycerol, or came from glycerol. And you can see, you're starting to see all the oxygen, so they're polar. And then there's a phosphate. That's why it's called a phospholipid. And then at the top, there's a serine, actually an amino acid, attached to it. So this is called phosphatidylserine. And there's a number of different ones that have different amino acids or different groups at the top in blue. Now, if we look at cholesterol, cholesterol is a much smaller molecule. Also has a hydrocarbon tail, so it's amphipathic, and the only polar portion is this single OH group. And it also has a series of ring structures, which makes it kind of a flat molecule. And it ends up inserting into the membrane. And then we have the glycolipids, also two nonpolar hydrocarbon tails, and then they are attached again by kind of this glycerol molecule, which is CH2. And then what's attached on there, there's an oxygen, which is polar, and then it's showing on the very right-hand side a galactose. So these are sugars which have lots of oxygens. So these are the 3 types: phospholipid, cholesterol, and the glycolipid. Now these lipids, the phospholipids and the glycolipids and cholesterol, can move around in the membrane. They're not fixed. And so this next slide just kind of gives you an idea, and then we'll talk about some experiments later. So what this says is the membrane phospholipids move within the lipid bilayer. They can float. I mean, you can think of it almost as a sea. It's a surface that has lots of movement. So they can spin, they can rotate, they can kind of jiggle back and forth, they can exchange with each other. That's called lateral fusion. And what they say flip-flop, meaning an exchange of one on the bottom, one on the top, it rarely occurs. What really happens is this kind of lateral fusion. So when you think of a lipid bilayer and a membrane, just think of just like floating. They're actually floating. Now, when you introduce cholesterol in there, there's what's showing here is phospholipids, and also the glycolipids, you can imagine, in there. But when you introduce the cholesterol, which makes up 10% of the lipid bilayer, it causes these lipids to become more fixed or more rigid. So that's what's shown here. Is so cholesterol, the introduction of cholesterol tends to what they call stiffen the cell membrane. So it makes it a little bit less fluid, a little bit less movement. And presumably that's one reason why if you have enormous amounts of cholesterol, the membranes can actually get a little bit brittle. But the function of the cholesterol is to kind of help stabilize these membranes. Now, just lipid bilayer alone, without any proteins, just talking about the lipid bilayer, it is an absolutely amazing barrier or boundary for the cell. And that's because of the properties of the internal hydrophobic region of the lipid bilayer. That a lot of molecules that are charged or are polar cannot make it through. So it acts as a really, really amazing boundary for the cell. So here we're looking at a diagram which kind of describes which molecules can flow through the membrane and which cannot. And again, this is based on all this is based on the hydrophobic interior of the membrane. So small nonpolar molecules can easily pass through the membrane. They have no charge. They can make it through the internal nonpolar region. So oxygen, CO2, nitrogen can all get through super easily. Steroid hormones are also nonpolar. They can get through the membrane. And we actually talked, I kind of alluded to this earlier on, because when we talked about steroid hormones in terms of transcriptional regulation, like the estrogen receptor, the receptor was in the cytoplasm, not on the cell surface, because these things get right through the membrane, and so the receptor's in the cytoplasm. Then we move down to small uncharged polar molecules. They're polar, they're not charged, and they're small, like water. So water can get through the membrane. It can work its way through the membrane. That's what's thought, because of the fact that it's tiny. Glycerol and ethanol, they can't really get through the membrane. They're a little bit too big, and they have they're a little bit more polar. Water can get through to some degree, but in addition, there are proteins that act as pores that specifically let water through, and those are called aquaporins. Aquaporins. And we'll look at the structure of one of those a little bit later. But there are proteins that allow water through, even though it can't get through on its own to some extent. Now we talk about the larger uncharged polar amino acids, polar molecules like amino acids, glucose, and nucleosides. They have a little tiny red arrow. I would say that none of these can get through the membrane. I don't think that red arrow should be there. So for all practical purposes, amino acids cannot get into the cell. Glucose cannot, nucleosides cannot get into the cell. They're way too polar. And then you have fully charged, like ions, even though they're tiny. Hydrogen, sodium, potassium, and so on. Those are fully charged. They cannot make it through the interior hydrophobic. They just can't get in there. So you see how just the nonpolar nature of the interior of the membrane makes this thing an excellent boundary just on its own. It separates the exterior of the cell and the interior of the cell very well. Now, that's about all I really want to say about the lipid bilayer per se and what it does. But you can see it actually is quite important. But 50% of the 50% of the membrane is made of protein. And that's really what carries out all the really complex activities of what happens with respect to the membrane. So 50% of the membrane is protein. And we're going to spend time looking at a lot of different proteins and what they do. But remember, when we're talking about these membrane proteins, we're talking about membranes. So it just shows here there's channels and transporters that let things in and out of the cell, like glucose, which cannot get into the cell. There's specific proteins that allow glucose to get in. Certain things can't get out because of the charge. There's certain proteins, channels, or transporters that let them out. There's what they call anchors. These are proteins, we'll talk about those later as well, that allow cells to interact with each other. There's receptors, which are extremely important. They recognize signals such as hormones, growth factors, even sometimes sugars or amino acids will act as triggers. And the receptors send signals into the cell for the cell to carry out specific activities. So for example, when you take a resting cell and treat it with a growth factor, the growth factor does not enter the cell. The growth factor binds to a receptor on the cell surface, and then it goes through an allosteric regulatory change, and then some activity occurs in the cytoplasmic side. And then it shows, and we'll talk about those as well. And then there's enzymes which can be present attached to the membrane that carry out enzymatic activities like kinases and so on. Now these proteins, just like the lipids which can move around, the proteins are free to move too. Not always. Let's change that. Many proteins can move around in the cell or in the membrane. Not all. So if you look at this figure, it shows the anchors. There's other proteins that attach to them. That's shown in brown and yellow. And so those tend to be less mobile because they are attached to other proteins in the cell. They can be part of the cytoskeleton. So they're kind of fixed. But receptors and these channels and these enzymes, all kinds of proteins, are free to move around in the cell. And I'll just show you a classic experiment which demonstrated that. Now this is an experiment using cells. In fact, they made you can take cells, and with certain compounds you can get the cell membranes to fuse. And when you get those cell membranes to fuse, they're called cell hybrids. And in this case, they're using a mouse-human cell hybrid. So the mouse cells have proteins on the cell surface. The human cells have proteins on the cell surface. And what happened here in this particular experiment, they took antibodies that recognized proteins on the mouse cell and labeled those antibodies with rhodamine, which is a fluorescent dye which fluoresces red under UV light. So the mouse cells were labeled with a red marker. The human cells were labeled with fluorescein labeled proteins, and so those come out. They show it as blue, but it's actually more green. And so you can see the representation of the proteins on the cell surface by the different colors. And you look at under a fluorescent microscope, and you can see these things. So then they carried out the cell fusion. They get the cell hybrid, and it's a bigger cell, but it's a hybrid between the two. And you look at that immediately after the hybrid is formed. So they have time at 0 minutes after cell fusion, and you look at the distribution of the colors, and you can see that they're separate because one represents the mouse, one represents the human. But if you incubate these cells at 37 degrees, which is basically body temperature, and you let them go for 40 minutes, an hour, 2 hours, however long, and you look at that again, what you see is there's a complete and random distribution of the colors along the surface. And so the conclusion is that the membrane proteins are free to move. This is actually referred to as fluidity of the plasma membrane. Fluidity of the plasma membrane represents the fact that the lipids can move freely and the proteins, most of the proteins, can move freely. Now, how are these proteins attached to the cell? They're attached in a number of different ways, but the most common way, I should say, and many of the proteins that we'll be talking about are called integral membrane proteins, or sometimes referred to as transmembrane proteins. Actually, that's the word I use most, is transmembrane proteins. So how these the transmembrane proteins are part of the membrane. They're integral into the membrane. So let's take a look at what those look like. So transmembrane proteins have what are called transmembrane domains. Transmembrane domains. They can have multiple transmembrane domains. I'll show you pictures in a little bit. But if you look at the top, I just have a kind of a linear drawing of a protein with a transmembrane domain, which is shown in blue. So you have the amino terminus, carboxy terminus, and in the center, there's the transmembrane domain. The transmembrane domains are hydrophobic or nonpolar alpha helices. So they're the alpha helices that we talked about when we talked about protein structure, but all of the amino acids are nonpolar. And they are allowed then to integrate into the nonpolar interior of the membrane. So that's what's shown on the bottom left. So you see the lipids, and then you see the nonpolar alpha helix in green, and it is in the membrane because it is stable in that nonpolar center of the membrane. So that's an integral transmembrane protein, and there are many different types. There are what are called single pass, which means that there's a single transmembrane domain, or there can be a multi-pass protein with multiple transmembrane domains. So for example, the sodium channel, which we'll talk about later when we talk about transport of ions in and out of the cell, the sodium channel has 24 transmembrane domains. And I'll say, the fact that integral or transmembrane proteins have these transmembrane domains makes them easily identifiable if you look at the amino acid sequence of the protein. So you can model, by X-ray crystallography, let's say you can model the protein, you can see these, let's say, you can see these alpha helices, and if you look at the sequence and see that they are all nonpolar R groups, then you can kind of assume that those represent transmembrane domains. So you can identify at least, proteins that may be on the membrane if you're trying to figure out their function. So this is just an example just to show you, these are all transmembrane proteins, and they can be really super complex. So you have, there's the light harvesting complex 2, which is part of photosynthesis, many transmembrane domains. Of course, that's in the thylakoid membrane. Then you have, let's see what else, aquaporin. Aquaporin is what I just mentioned to you, is a protein that allows the transfer of water. And it just gives you an example of how these things end up in the membrane because they have all of these different transmembrane domains, multiple transmembrane domains. So the bottom line is that integral transmembrane proteins all have these transmembrane domains, and they are nonpolar alpha helices. There are other ways for proteins to be associated with the membrane. We'll look at that here next. So membrane proteins can associate with the lipid bilayer in different ways. Okay. But the way I want to point out, aside from the transmembrane domains, and also if you have the integral membrane proteins, which is shown in the center, it says monolayer associated. These are basically the same. Okay, it's a nonpolar transmembrane domain, and then you have other ones. Let's say under D on the right-hand side, you have the transmembrane domains, but then you have proteins attached to those proteins. So those would be referred to as peripheral transmembrane proteins. They're really not transmembrane. They're really not membrane proteins, but they're associated with the membrane. But what I did want to point out, which I show with the arrow, is the lipid-linked associated proteins. These are membrane-associated proteins, and the way they are attached to the membrane is because the lipids have this, they're amphipathic, and they have a nonpolar tail or nonpolar region, which then integrates into the membrane because it is stable in the interior of the nonpolar interior of the membrane. So you can have transmembrane proteins, and you can have lipid-linked proteins associated with the membrane, and then you can have these peripheral membrane proteins. Okay. So I just want to continue a little bit on the transmembrane proteins, and the question is, how do they get there? How do they get into the membrane? They get into the membrane because when we talked about the co-translational transfer, and that was where the ribosomes were attached to the ER membrane, and then there was a signal sequence that allowed the protein to enter the membrane through that channel. And then I showed you in one particular figure that, and I'll show you that figure again, that the membrane proteins are also introduced in that fashion, and that they stay in the membrane, and then they move up through the ER membrane by transport vesicles up to the Golgi, and then up through the clathrin-coated vesicles, and then they make it to the surface, and then they end up as receptors or signaling proteins or so on. So it turns out that these nonpolar alpha helices, in addition to functioning as a mechanism for maintaining the protein in the membrane, they also function as additional signal sequences that allow the protein to stop transport. I'll show you that in a second. So this was the drawing I was referring to that I talked about and I showed you earlier. I want you just to look at, so there's the ER, there's the Golgi, and there's the cell surface. It's showing cell surface protein, which I have drawn as kind of a Y. That may be a receptor. That obviously would be a transmembrane protein with a transmembrane domain, which would be alpha helix, nonpolar alpha helix. And these things enter the system by the same co-translational transfer. But the nonpolar alpha helix acts as a stop transfer signal. And so they end up stuck in the membrane. And so I just want you to look at that a little bit carefully and see how this kind of works. So you have a protein, which let's say is a receptor. Let's look at the very bottom at the ER. The receptor portion is on the interior of the ER, in the lumen of the ER. And of course, that's getting glycosylated as it moves along. It's in the interior. And then it gets packaged into the transport vesicles, it gets transported into the Golgi, all the time attached to the membrane. And then it moves up into the clathrin-coated vesicle. And you look at that very last vesicle up at the top, you see because the membrane opens up, it inverts, in a sense, inverts the receptor or the protein so that now the receptor portion, that Y portion, is now exposed to the cell surface, but it's glycosylated. And the interior, what's in the interior of in the cytoplasm is not glycosylated because it was never exposed to the lumen of the ER or the Golgi. So these transmembrane parts of the protein, these transmembrane domains, act as additional signals, and that's shown in this next slide here. So they act as start and stop signals. If we look at the line drawing, so the amino terminus, carboxy terminus, the red portion is the ER signal sequence. That is referred to as a start transfer signal because it starts the transfer during the co-translational transfer process. And then going along there, the blue is the transmembrane domain, which is the nonpolar alpha helix. This is referred to as a stop transfer signal. Stop transfer. I'll show you a multi-pass membrane protein in a second. Okay, so looking at the bottom basically just describes what happens. So you have the beginning of it's not showing the ribosome, but you're showing the co-translational transfer taking place. So of course the red ER start signal gets passed in through the protein translocator. It ultimately gets cut off by the signal peptidase. But as translation continues, it pushes the protein through this protein translocator until it gets to the hydrophobic alpha helix, which in this situation is a stop transfer sequence. It signals it somehow to stop transferring. Maybe it gets stuck in. I don't know how it works, actually. Not sure anyone really does. But the observation is that it stops transfer, and then anything that's synthesized after that stop transfer will be on the outside in the cytosol. It won't get transferred in. So that's what's showed on the very right-hand side. So you see the amino portion is in the interior of the ER lumen. That got transferred in until it hit the alpha helical transmembrane domain, and it stopped transferring, and then everything that's made after that is on the outside in the cytoplasm. So this would be a single pass transmembrane domain, and that would be moved up the system, maybe to the cell surface. Now, the multipass transmembrane domains have multiple nonpolar alpha helices, and they work as stop and start transfers sequences in sequence. So the first one would be a stop, the next one would be a start, so start transferring again, third one would be a stop, and so on. So here I have a drawing just to kind of illustrate that. So here we have just an example of a multi-pass. This would be 3 transmembrane domains, and what I have written here is their alternate start and stop transfer signals. Each one's in a transmembrane domain. So at the beginning, at the amino terminus, you have the ER start, and then they go in order. So the next one was stop, the next one was start transfer again, and the next one is stop. And so if you look at the bottom, it kind of shows, in a sense, what it would look like. The lumen is at the top. It's different than what was shown before. They presented it differently. So the lumen of the ER is at the top, and the cytoplasm would be on the bottom. And so you have the start transfer by the ER signal sequence, and so it transfers through. Then you hit the first stop, it holds it. Everything after that is on the outside, so that's why it's showing it a little bit on the cytoplasmic side. And then the next one that comes along is a start transfer signal. So you have transferring a lot because you see there's a larger separation between the start and that last stop signal. And so you get a larger loop going in, and then it hits that next one, which is a stop, and then you get a little bit of the carboxy terminus on the cytoplasmic side of the membrane. Okay, so that's basically structure of the membrane and the overview of the types, not the types of proteins. Well, yeah, we talked about the types of proteins, but an overview of what these proteins look like in the membrane, and the fact that they're mobile in the membrane, and their basic structure in the membrane, and what these transmembrane domains look like. Now we're going to start talking about some of the activities of what these proteins do. That will be in the next presentation.